Also going to be two specific functional groups that are always
In
attached
this lesson,
to that alpha carbon.
we will talk about what are proteins,
One of those will be the amino functional
what is a
group.
spectrophotometer,
That is highlighted here in
what
green.
is the Bradford assay,
And the other
and how
functional
can
group
it be
is going
used
to
to
be a
quantify the
carboxyl
amount
group.
of protein in
That
solution.
group is highlighted here in yellow.
We will talk about what are dilutions,
Also
both simple
attached to the
and
alpha
serial
carbon,
dilutions,
we will have a
and how they are
generic
calculated.
group, which is called the R group.
And then lastly,
and sometimes
we'll
it's
talk
referred
about
to
some
as a side
specific
chain.
tips that you can use
That
to
here
successfully
is highlighted
complete
in
this
blue,
lab.
and that is the variable part of the amino acid. So different amino acids will have different R groups, but they're all going to have that alpha carbon, the amino functional group. They have a lot of unique structures to them. This slide contains all the amino acids that have nonpolar side chains. Even though the structures are variable, smaller building blocks, all of these molecules contain an alpha carbon. On this slide, we have a generic structure highlighted of an amino acid, and they're attached to that alpha carbon features. They all have one amino group, which is being shown in the very center of an amino acid, and they have one carboxyl central carbon atom, which is being shown which we refer to as the alpha carbon. Also attached to that alpha carbon is shown we have the R group on this or the side chain. And that is being shown. There's also going to be two specific functional groups that are always attached to that alpha carbon. Polar or one of those will be the amino functional group, as we have with the previous amino acids here in green. All of these have an alpha functional group is going to be a carboxyl, a red carbon atom. That group is highlighted here in yellow. Contains an amino group highlighted also in green. Attached to the alpha carbon, we will have a generic highlighted in yellow, which is called the R group, and sometimes it's referred to as a side chain. Again, notice that all of the R highlighted in blue are different, and that is the variable part of the amino acid. Two amino acids, so can different be amino acids by a will have different R groups, but they're all going to have that alpha carbon, that the amino functional carboxyl group, and the carboxyl amino acid group, and the amino group. This slide contains all the amino acids that have non-polar side chains. That is part of the carboxyl group. Even though the structures are variable, all of these molecules contain an alpha carbon, the amino center, which has been highlighted in red. Attached to that alpha carbon, they all have one amino group, which is being shown in green. The OH group and they have one carboxyl functional group, will combine which is being to form one water molecule. Also attached to that alpha carbon, will then be joined together the R group by a covalent or the side chain, and that is being shown in blue. Click on this slide, we have the amino acids that contain polar or charged side chains constructed by joining many as we have with the previous amino acids. Peptide bonds. All of these have an alpha carbon example shown in the center as a red carbon atom, and in this case, that alpha carbon contains highlighted an amino group so that highlighted in green, a carboxyl group amino acids highlighted in yellow, connected together to make and then an R group highlighted in blue. Again, notice that all of the R groups are different from each other. Before we introduce the spectrophotometer, two amino acids can be connected together by a peptide concerning light. The peptide bond is a type of electromagnetic radiation between the carboxyl group this light of one amino acid and the amino group. The light that we see is made up of many different colors. Click on the OH colors that is part of the carboxyl group of alanine. Solutions and compounds on one of the hydrogens that is part of the amino group. This results in the colors that we see around us. The colors that we see are colors that are being reflected. It is a dehydration reaction. The OH group, the primary and the hydrogen that you will use for this will combine is to form one water molecule. This instrument the two amino acids by measuring will then the light be joined together absorbed by a covalent bond. Keep in mind that light is made up of many different colors. Click on and the colors that we view are the colors that are reflected. Polypeptides. So when we talk about the spectrophotometer, this instrument is actually measuring the colors that are not seen. Here we have an example of a small polypeptide. Another way to look at it and in this case, is that this instrument compares the light that goes into a sample versus the light that comes out of the sample. How many amino acids have been connected together to make this polypeptide? We look at the result as absorbance. This figure illustrates how the spectrophotometer works. Before we introduce the spectrophotometer has a light source that will emit all colors or wavelengths of light. Within the machine, there is a wavelength selector, and that will allow only a specific wavelength of light to pass through. Is made up of many the machine allows you to select which wavelength this will be. That wavelength that is selected absorb light will be emitted towards your sample. This results in the colors that we see around us. Some of it will pass through. The colors that we see are colors the light that is absorbed is called absorbance, and the light that passes
The primary
through
instrument
is called
that you will use for
transmittance.
this lab
is the
there will be a
spectrophotometer.
detector on the side of the sample
instrument
opposite
works
the
by
light
measuring
source,
the light
and
that
that
is
will
absorbed
detect
by a
the
sample.
transmittance. Keep in mind that light is made up of many different colors, and the colors that we view are the colors that are reflected. So when we talk about the spectrophotometer,
In this lab,
you will be using the Bradford
actually
protein
measuring the
assay
colors that
to
are not
quantify
seen.
or measure the amount of
Another
protein
way to look
that
at
is
it
in solution.
is that this instrument
This assay
compares
is
the
based
light
on
that
different
goes
forms
into a
of
sample
Kumasi
versus
Brilliant
the
Blue
light that
dye.
comes out of the sample.
This dye molecule is
Typically,
a red-brown color
we look at the
in
result
the absence of protein
as absorbance.
When there is protein in
This
solution
figure
with
illustrates
this
how the
molecule, this
spectrophotometer
molecule
works.
is a blue color.
The spectrophotometer
So
has
as
a light
the dye
source
binds
that
more
will
and more
emit
protein,
all colors
it becomes
or wavelengths
more
of
and more
light.
blue. Within the machine,
shift
there is
in
a
color
wavelength
of the
selector,
Kumasi blue
and that will
molecule
allow
in
only
the
a
presence of
specific
protein
wavelength of
is
light
visible
to pass
to the
through.
naked eye.
The machine
However,
allows
with the
you to
use of a
select which
spectrophotometer,
wavelength
we
this
can get a
will
more
be.
accurate idea of the
That
extent
wavelength that is
of this
selected
color change.
will be emitted
So
towards
in the
your
absence
sample.
of protein,
Some of it will be
Kumasi blue
absorbed,
is a red-brown
some of
color,
it will pass through.
and it tends to absorb light.
The light
at
that is absorbed is called
465
absorbance,
nanometers.
and the light that passes
In the presence
through
of protein,
is called transmittance.
this molecule becomes a blue
There will
color,
be a detector
and it absorbs
on
light
the side of the
at
sample opposite the light
595
source,
nanometers.
and that will detect
So
the
as the amount of
transmittance.
protein in solution increases, the absorbance at 595 nanometers also increases. A standard curve
In
is
this
a graphical
lab, you will be
representation
using the
of
Bradford
the
protein
absorbance of the
assay
Kumasi
to
blue
quantify
dye at
or measure
595
the amount of protein
nanometers
that is
when
in
it is in the
solution.
presence of various
This
concentrations
assay
of protein.
is based on different forms of Kumasi
This standard
Brilliant
curve
Blue
is useful
dye.
because once
This
we have
dye
the curve
molecule
drawn,
is a
we can
red-brown
predict
color
the
in the
concentration
absence
of
of
protein
protein.
in a solution
When
based
there is
on the
protein in
absorbance
solution
value
with
that it
this
produces.
molecule, this molecule is a blue
For
color.
example, suppose
So
that we
as
have a
the
sample
dye
that
binds
has
more and more
produced an
protein,
absorbance
it
of
becomes
0.4.
more and more blue.
Click the
The
point
shift
on the standard
in color
curve that
of the
represents
Kumasi
an
blue
ABS
molecule
595
in the presence of protein
0.4.
is visible to the naked
To
eye.
identify
this point,
However,
we would
with the
first
use of a
locate an absorbance of
spectrophotometer, we can get a more
0.4
accurate
on the
idea
y-axis.
of the extent
Once
of
we've
this color
found that,
change.
we move horizontally
So in the
across
absence
and
of
we mark
protein,
that point on our
Kumasi
standard
blue
curve.
is a red-brown color.
Once we
and it
know where
tends to
that point
absorb light
is, we
at
can predict the amount of
465
protein in that
nanometers.
specific tube.
In the presence of
To predict the protein,
this
we
molecule
would drop a
becomes
line
blue
down
color,
vertically
and it
from that
absorbs
point,
light at
and we would figure out what the
595
x-axis value for that
So
point would
as
be.
the amount of protein
In this
solution
case,
increases,
it is approximately
the absorbance at
0.16
595
milligrams per
nanometers
milliliter.
also increases.
So that value there tells us approximately
A
how
standard
much
curve
protein
is a
we
graphical
have in that tube
representation
that produced
of
that
the
absorbance
of the
of
Kumasi blue
0.4.
dye at 595 nanometers when it is in the presence of various concentrations of protein.
On a graph, there is both
This
a
standard
y-axis
curve is useful
and an
because
x-axis.
once we have the curve
When
drawn,
we draw a standard
we can
curve
predict the
for the Bradford
concentration
assay,
of protein
we
in a
always put
solution
the absorbance
based
at
on the absorbance
595
value that it
nanometers
produces.
on the y-axis. The
For
y-axis
example,
is the vertical
suppose
axis,
that we have a
and it
sample
is shaded
that has
or
produced
highlighted
an
here
absorbance
in blue.
of 0.4.
On the x-axis,
Click the point
we would
on
put
the
our
standard
protein
curve that
concentration,
represents an ABS
and the x-axis is
595
a horizontal
of
axis,
0.4.
and this is shaded here
To identify
in green. this point, we would first
The curve that we
locate an
see drawn
absorbance
on
of
this graph
0.4
is made
on
up
the
of
y-axis.
an infinite number of points.
Once we've found
Just a
that,
few of
we
those
move
points have
horizontally
been marked
across
here for
and we mark
illustration
that point
purposes.
on our standard curve.
Each point that we have
Once we
on
know
this
where
curve
that point
made
is,
up
we
of a
can predict the
y-axis
amount of
value
protein in
and
that
an
specific
x-axis
tube.
value. For
To
example,
predict the protein,
if we
we
take
would
one
drop
of
a
those
line
points,
down
we can
vertically
move
from that
horizontally
point,
across,
and we would
can
figure out
the
what the
y-axis
x-axis value for that
or
point
we
would
could go
be.
vertically down from
In
that
this
point,
case,
and we
it
could
is
figure out
approximately
the x-axis
0.16 milligrams
So,
per
for
milliliter.
example, if we look
So
at
that
this
value
specific
there
point
tells
here,
us approximately
it has
how
a
much
y-axis
protein
value
we
of
have in that tube
0.84.
that produced that
So that would be
absorbance
the
of
absorbance value
0.4.
of that point, and it has an x-axis value of 0.34 milligrams per milliliter.
On a graph, there is both a y-axis and an x-axis. When we draw a standard curve for the Bradford assay, we always put the
standard
absorbance
curve
at
is very useful
595
for taking a
nanometers
solution
on the
that has a
y-axis.
specific absorbance,
The y-axis
and predicting
is the
how much
vertical
protein
axis,
is in that
and it is
specific
shaded
sample.
or highlighted here in blue.
Consider that you have a solution with an
On the
absorbance of
x-axis,
1.9.
we would put our protein
What would be the
concentration,
protein concentration
and the
of this
x-axis is a
solution?
horizontal axis.
The standard
and this
approach
is
for a
shaded
question
here
like
in
this
green.
would be to find 1.9
The curve that we see
on
drawn
the
on this
y-axis
is made
and
up
then
of an
locate
infinite
that point
number
on
of
the
points.
curve.
Just a few of those points have been marked in this example is that region of the curve each point is very flat on this curve is made up of a y-axis value exactly and an x-axis 1.9 would fall on this line. If we take one of those multiple locations, we can move where that horizontally across, and that we can figure out the y-axis value, corresponding x-axis value, or we could go vertically down from that point. So we reach kind of a plateau here on our curve, which makes it very difficult to use that specific point to predict the concentration. It has a y-axis value of 0.84. So that would be the absorbance value of that point. A linear portion, and it has an x-axis value, and we have a 0.34 milligrams per milliliter. The most useful region of the curve is going to be the linear region. If we have an absorbance value that falls within that linear region, the standard curve is very useful to mark taking a solution on the curve specific and then figure out the protein concentration and predicting how much protein is in that specific sample. Consider that you have a solution with an absorbance of 1.9. Since linear region is the most protein concentration of this solution, we want to have a range of protein concentration for a question like this to ensure that we do find 1.9 absorbance value on the y-axis within that linear and then locate that point on the curve. If we have a value that falls on that area in this example, we can be sure that we can establish what the corresponding x-axis value of that point should be. This makes it difficult to determine exactly where 1.9 would fall on. In other words, we do not know what the multiple locations of that where that point could be. We want to go ahead and establish what range of dilution x-axis of that sample, so that we can be sure that we will obtain at least one of them curve that has a value that falls within that area of the region to predict the concentration in an unknown solution. So we have, with our standard curve, a linear portion diluted to prepare solutions that are less concentrated than the original. The most useful region of the concentration is going to be the linear region. In those dilutions, if we have an absorbance value that falls within that linear region, calculate those. It will be very easy for us to mark the equation that we would on the curve if we had figure out the protein concentration that we wish to make in that solution. Is listed here. In this equation, solution 1 is the original linear region is the most useful that we are working of the curve. We want to have a range of protein concentrations more diluted to ensure that we do obtain an absorbance value that falls within that linear portion. The V's in this equation represent volume. If we have a value that we're going to take from the original solution, we can be sure that we have a volume establish what the corresponding x-axis value of that point should be. And then the concentration is represented by C. An unknown sample, we know what our original solution concentration is, so that would be sample will be. And then we go ahead and establish a range of dilution C2 of that we wish to make up, so that we can be sure that we will obtain at least one of them that has a value that falls within that area of the curve. A 4 molar solution to prepare 10 milliliters of a 0.2 molar solution. A solution can be diluted to prepare solutions that are less concentrated than the original. If we have specific concentrations that we wish to prepare in those dilutions, then we can use a very simple equation to calculate those. One type of dilution is what we call a simple dilution. The equation that we would use to calculate a simple dilution, we would take the volume of the sample and we would divide that by the total volume, including the sample plus the buffer. Original, more concentrated For example, that we suppose that you have 2 milliliters of a sample that is combined with 18 milliliters of buffer that we wish to prepare. To calculate this dilution, we would take the volume of the sample, which is 2, so we have and we would divide that by the total volume, which is 2 and we have a volume that we wish to make the buffer of the second solution. This simplifies to 2 over 20, represented by and we can reduce that down to 1 over 10. We know what our original solution so this dilution would be 1 over 10. And then we have another way to look at dilutions is to use what we call a dilution factor of our new solution. This is essentially the reciprocal. We take the total volume of the sample plus the buffer and we divide that by the sample. 10 milliliters if we applied this to the same example, we would have a dilution factor of 10. Serial dilution is the stepwise simple dilution of a substance by a constant dilution factor. In this case, the source of the dilution material for each step comes from the diluted material One type of the previous step. Is what we call a simple dilution. As you can see in this illustration, a simple dilution, with each step of serial dilution, the sample becomes more and more diluted. And we would divide that by the total volume, including the sample plus the buffer. For example, if we are preparing cereal dilution,
that you have 2 milliliters of a sample,
we go ahead
that
and
is
fill a number
with 18 milliliters
with the amount
of buffer that we are going to be using.
To calculate this dilution,
in this example,
we would take the volume of the tubes,
which is 9 milliliters,
and we would divide that by the total volume.
We would then take
2
sample
plus 18
from the original
if
tube,
we include the buffer.
in this case 1 milliliter.
This simplifies,
and we would add it
to
the first
over
test
20,
tube.
and we can reduce that down to 1 over 10.
Once we've prepared this first dilution,
so
we
this
can calculate the dilution
would be 1
factor.
over 10.
To calculate the dilution
Another
factor,
way to look at dilution,
the total volume
is to
of
use
the
what we
sample
call a dilution
plus the
factor.
1
This
plus
is
9
essentially
in this
the
case.
reciprocal,
and we would
we
divide
take
that
the
by
total
the volume
volume
of the sample
plus
which
the
is
buffer,
1 milliliter,
and we divide
in
that
this
by
case.
the sample.
That gives us a
If we
dilution
applied
factor
this to
of
the
10,
same example,
and that
we
is
would
commonly
have
written
a dilution
as
factor
10x.
of 10. If we look at our second
Cereal
tube,
dilution is
we
the
would take
stepwise
1
simple
milliliter
dilution of
from
a
the
substance
10x
by a
dilution,
constant dilution
and we would
factor.
move that to the next tube.
In this case, the source of the dilution
case,
material for
we would
each
dilute
step
that
comes from the
10
diluted
times
material
more,
of the previous
and so we
step.
would have 10 times 10,
As you can
which
see
is
in
going
this
to give us a dilution
illustration,
factor
with each
of
step of
100x.
cereal dilution, the sample
would repeat
becomes
this
more and
procedure
more dilute.
for as many tubes as we wanted, and notice that each time
If we are preparing cereal
exponentially
dilutions,
increasing
our
typically
dilution
we
factor.
go ahead and fill a number of tubes with the amount of buffer that we are going to be using. In this example, we would fill all of the tubes with 9 milliliters of buffer. Now let's put it all
We would
together
then take
in an example
sample
that
from
includes
the
the
original
Bradford
tube,
assay,
in this
and
case,
the
1
dilution
milliliter,
factors, and we would add it to the first test
Suppose
tube.
that a sample with a dilution
Once
factor
we've
of
prepared
15
this first
produces
dilution,
an
we
absorbance
can calculate
of
the dilution
0.5
factor.
when mixed with Kumasi
To calculate
blue,
the
which is
dilution
the
factor,
Bradford
we
reagent.
would take the total volume
And
of
we
the
want to
sample
know
plus the
ultimately
buffer,
what is the protein
1 plus
concentration
9
in
of
this
the
case,
original
and we would
undiluted
divide that
solution
by the volume of the sample, which is 1
The
milliliter.
first step
in this
question
that
is to
gives us
figure
the
out the
dilution
protein
factor of
concentration
10, that we
and
have
that is commonly
in the diluted
written
tube
as 10x.
that produce that absorbance of 0.5.
If we look at our second tube, we would
Based
take
on the
1
standard
milliliter
curve,
from
we've
the 10x
estimated the
dilution,
protein concentration
and we would move
in the
that
diluted
to the
tube
next
to
tube.
be 0.2 milligrams per
In this
milliliter.
case, we would dilute that
The
10
second
times
step
more
for a question like
and
this
so we would have
would
10
be to
times
determine
10,
the dilution
which
factor
is going to give
of
us
that
a dilution
diluted
factor of 100x.
This information is given to
We
us
would repeat
in the
this
question,
procedure
and we can
for
see
as
that we
many
have a
tubes
dilution
as
factor
we
of
wanted.
15.
And notice that each
Once
time
we know
we are
the protein
exponentially
concentration in the
increasing
diluted
our
tube,
dilution
and we
factor.
know the dilution factor, now we're ready to figure out the original protein concentration in that undiluted tube. So step number 3 is to multiply the protein
Now let's put it all
concentration in the
together
diluted
in an
tube
example
by the
that
dilution
includes the
factor.
Bradford assay
If we
and the
do
dilution
this,
factors.
we come up with an original protein
Suppose that a
concentration
sample
of
with a
3
dilution
milligrams
factor of
per
15
milliliter
produces an absorbance of 0.5 when mixed with Kumasi blue, which is the Bradford
Suppose that a
reagent.
sample with a dilution
And we want
factor
to
of
know
20
ultimately
produces an
what is the
absorbance
protein
of
concentration
0.24
of the original
when mixed with
undiluted
Kumasi blue,
solution.
the Bradford reagent. What is the
first step
protein
in this
concentration
question
of the
is to
original
figure out the
undiluted
protein
solution?
concentration that we
The
have
first step
in the diluted
is to
tube
figure out the
that
protein
produced that
concentration
absorbance of
in the diluted
0.5.
tube.
To
Based
determine
on the
this,
standard
we
curve,
need to look at a
we've
standard
estimated
curve
the protein
and
concentration
figure out
in the
what is the
diluted
protein
tube to be
concentration
0.2
of a tube
milligrams per
that produces
milliliter.
an absorbance of 0.24.
The second step for a
based on
question
the
like
standard
this
curve would be to
the diluted
determine the
tube
dilution
has a
factor
protein
of that
concentration
diluted tube
of 0.1
This information
milligrams
is
per
given
milliliter.
to us in the question.
The second
and we
step
can see that
is
we
to
have a
determine
dilution
the dilution
factor
factor
of 15
of the diluted tube.
Once we
In
know
this
the
case,
protein
that
concentration
information
is
is presented
presented
in the question,
and we know the dilution
and we can see that
factor,
we have a dilution
now we're
factor
ready to
of
figure
20.
out the original protein
In step
concentration
number 3,
in
we
that
put it all
undiluted
together,
tube.
and we're going to
So
multiply
step
the
number
protein
3
concentration
is to multiply
in
the
the
protein
diluted tube
concentration
by
in
the
the
dilution
dilution
tube
factor.
by the dilution
In this
factor.
case, we multiply
If we
0.1
do
milligrams
this,
per
we
milliliter
come up with an
times
original
20,
protein
and
concentration
that's going to give
of
us a
3
final
milligrams
protein
per
concentration of 2 milligrams per milliliter. Suppose that a sample with a dilution factor of 20 produces an absorbance of 0.24 when mixed
Before
with
arriving
Kumasi
to lab,
blue.
the
You
Bradford
should complete
reagent.
all calculations
What is
for
the protein
tables 1 and 2
concentration
in your
of
lab
the original
manual.
undiluted solution?
The first step is to figure out the protein concentration in the diluted in a number of unknown solutions. To determine this, it is important that standard measurements be made as accurately as possible. What is the protein concentration of a tube that produces an absorbance, and part of your grade will be based on that. Based on the standard curve, as you mix the diluted samples, you want to take special care to ensure that there is no cross-contamination per milliliter. Once you have the second step, all of your protein to determine the dilution factor, take a look at your test tubes and make sure that as the protein information is presented in the question, the amount of blue, and we can see that we have a dilution factor also increases. If you find that this is not the case, we put it all together and we're going to multiply the protein concentration in the diluted tube by the dilution factor. For measuring your samples. In this case, we multiply 0.1 milligrams per milliliter times 20, and that's going to give us a final protein concentration inside your lab of 2 milligrams per milliliter. When you get ready to measure your samples, the samples do get put in a special holder called a cuvette. This is a square holder that will slip right down before arriving to lab. You should complete all calculations for tables 1 and 2 at the bottom in your lab manual. You will notice that there are different widths to the cuvette depending on its orientation. You will be determining the protein concentrations. It is very important that you make sure that the cuvette is in the proper orientation. It is important that all of the measurements be made as accurately as possible. This is because this will affect the final concentration or the way that it is oriented, and part of your grade will be based on that this instrument produces. As you mix up the samples, if you look at the yellow arrow to ensure that there is no cross-contamination, this shows the direction that the beam of light will be traveling all of your protein in the spectrophotometer. Take a look at your test tubes and make sure that as the protein concentration increases, when the cuvette is placed in the machine in the test tubes, you want to make sure that the light is going to travel through the greatest distance of the cuvette. Then you do want to repeat this step. After you read a sample, you will use a spectrophotometer. You will rinse the cuvette using the provided water bottles. After you have completed all of your dilutions and Bradford assays, so you will want to shake out any remaining water droplets before you add the next inside your lab manual. When you get ready to measure your samples, you will wash all test tubes and cuvettes with detergent using this is a square brushes to remove all traces of blue dye. Into the spectrophotometer. This is very important. And failure to do so will affect your grade for this lab. You will notice that there are different calculations to the cuvette depending on all other lab procedures and cleanup. It is very important that you make sure that the cuvette is in the proper orientation when you place it in the spectrophotometer. This is because this direction, or the way that it is oriented, will affect the reading that this instrument produces. If you look at the yellow arrow in this diagram, this shows the direction that the beam of light will be traveling in the spectrophotometer. The green box represents where the cuvette will be held. When the cuvette is placed in the machine, you want to make sure that the light is going to travel through the greatest distance of the cuvette. After you read a sample in the cuvette, you will rinse the cuvette using the provided water bottles. You will be reusing the cuvettes, so you will want to shake out any remaining water droplets before you add the next sample. When you have finished the lab, you will wash all test tubes and cuvettes with detergent using the test tube brushes to remove all traces of blue dye. This is very important, and failure to do so will affect your grade for this lab. The graphs and calculations should wait until all other lab procedures and cleanup have been completed.